Welcome to this lecture on the hormonal regulation of metamorphosis. Imagine living inside a house that needs to be completely rebuilt into a mansion, but you cannot move out during the construction. You would need a master contractor to perfectly time when to tear down the old roof and when to build the new walls, ensuring the house never collapses while you are inside. In the biological world, a tadpole turning into a frog faces this exact same monumental challenge. Today, we will uncover how chemical messengers called hormones act as these master contractors to orchestrate this incredible transformation. Building on that house analogy, let us look at how this construction project actually works in nature. The master contractors in this biological scenario are hormones. These are chemical messengers that travel through the bloodstream to deliver precise, time-dependent instructions to the entire body. During metamorphosis, these hormones have to multitask perfectly. They tell specific cells to self-destruct, just like the cells in a tadpole's tail, while simultaneously commanding other cells to multiply and form entirely new organs, such as lungs and legs. Understanding why this matters is crucial, because without this precise coordination, the animal would not survive the transition. Now that we understand the overall challenge, let us examine the specific hormones responsible in amphibians. The dramatic physical transformation from a tadpole to a frog is primarily driven by thyroid hormones. Specifically, the hormones thyroxine and triiodothyronine take the lead. These endocrine signals are responsible for triggering the resorption of larval organs, like the gills and the tail, while simultaneously promoting the development of adult structures. It is a highly precise, time-dependent process. If these hormones are released too early or too late, the developmental sequence breaks down, and the amphibian will remain trapped in its juvenile form forever. While amphibians rely on thyroid hormones, let us shift our focus to see how insects handle this exact same problem. In insects, the process of metamorphosis is controlled by a delicate, competing balance between two very different signals. On one side, we have the juvenile hormone, which acts as a chemical anchor to maintain the larval state and keep the insect young. On the other side, we have ecdysone, a hormone that actively promotes molting and adult development. The entire life cycle of a caterpillar turning into a butterfly depends on the biological tug-of-war between these two competing chemical messengers. So, we know which hormones are involved, but this brings us to an important question. How do they actually change the animal's body? These hormones function by physically entering the target cells and binding to specific intracellular nuclear receptors. Once the hormone is bound, this newly formed hormone receptor complex acts as a powerful transcription factor. It binds directly to the DNA inside the nucleus, acting like a master switch. By turning specific target genes on or off, these complexes rewrite the genetic instructions of the organism, orchestrating the massive physical transformation from the inside out. Before we move on to the mathematics of this process, let us see why this knowledge is so critical in the real world. Scientists and agricultural experts use our understanding of these hormonal pathways to develop highly targeted, environmentally safe pesticides. By creating synthetic chemicals that perfectly mimic insect juvenile hormones, we can spray crop fields or mosquito breeding grounds. This artificial signal prevents these pests from ever reaching their biting, reproducing adult stages. This incredible application of developmental biology saves millions of human lives from deadly diseases like malaria. And it protects our global food supply without harming other wildlife. Now that we understand the biological mechanism, let us look at the mathematics of receptor binding to see exactly how this works. A hormone floating in the blood does absolutely nothing unless it successfully binds to a receptor. We model this using the fractional occupancy formula. Y equals H divided by the sum of Kd and H. Here, Y represents the fractional occupancy, meaning the percentage of receptors that have a hormone bound to them, ranging from 0 to 1. H stands for the concentration of the active hormone in the blood, measured in nanomoles per liter. Finally, Kd is the dissociation constant, representing the hormone concentration where exactly half the receptors are occupied. If the hormone concentration increases, the fractional occupancy increases, driving a stronger biological response. Conversely, a high Kd means the receptor has a low affinity, requiring much more hormone to trigger metamorphosis. Let us apply this formula to a concrete example involving a tadpole in its early pre-metamorphosis stage. We are given that the dissociation constant for the tadpole's thyroid hormone receptors is 4 nanomoles per liter. During this early stage, the blood thyroid hormone concentration is very low, at just 1 nanomole per liter. We need to calculate the fractional receptor occupancy to see if metamorphosis will begin. We substitute the hormone concentration of 1 into the numerator and denominator and plug in the dissociation constant of 4. This simplifies to 1 divided by 5, giving a final answer of 0.20. Physically, this means only 20% of the receptors are occupied. This low level of activation is completely insufficient to trigger the massive genetic changes required, so the tadpole safely remains in its larval state. Following our previous calculation, let us see what happens when the tadpole reaches the peak metamorphic climax stage. The physical receptors have not changed, so the dissociation constant remains exactly 4 nanomoles per liter. However, the hormone concentration now spikes dramatically to 36 nanomoles per liter. We want to find the new receptor occupancy. Substituting 36 for the hormone concentration and 4 for the dissociation constant, our fraction becomes 36 divided by 40. This gives a final answer of 0.90. This indicates that a staggering 90% of the thyroid receptors are now bound to the hormone. This massive jump from 20% to 90% acts as a powerful biological switch. The overwhelming signal instructs the tail cells to self-destruct and the leg cells to grow, proving mathematically why metamorphosis happens suddenly as a climax. Now that we have modeled amphibian receptors, let us look at a different mathematical model used for insects. Because insect metamorphosis is controlled by 2 competing hormones, we define a metamorphic index, denoted by M. Conceptually, this formula represents the biological tug-of-war happening inside the insect's body. M is calculated by dividing the concentration of ecdysone by the concentration of juvenile hormone, or JH. Ecdysone is the driving force for maturation, measured in nanograms per milliliter. Juvenile hormone is the chemical anchor keeping the insect young, also measured in nanograms per milliliter. Because we divide these two, the units cancel out, leaving M as a single dimensionless number. If ecdysone increases, the index rises, pushing the insect toward adulthood. If juvenile hormone increases, the index drops, keeping the insect in its larval state. Let us test this new formula with a laboratory caterpillar. We're given that for this specific insect to successfully pupate and turn into a moth, its metamorphic index must strictly exceed a threshold value of 8. Currently, the caterpillar has an ecdysone concentration of 150 nanograms per milliliter and a juvenile hormone concentration of 25 nanograms per milliliter. We need to determine if it will transform. We substitute 150 into the numerator and 25 into the denominator. Dividing these gives a final answer of exactly 6. We must now compare this result to our required threshold of 8. Because 6 is strictly less than 8, the ecdysone signal is not yet strong enough to overpower the lingering juvenile hormone. Physically, this means the caterpillar will not pupate, but will simply shed its skin and emerge as an even larger caterpillar. To visualize everything we have calculated, let us look at this graph showing hormone levels over developmental time. The horizontal axis represents time, divided into pre-metamorphosis, pro-metamorphosis, and the metamorphic climax. The vertical axis shows the relative hormone concentration. Notice the blue curve representing prolactin, a hormone that promotes larval growth. It starts high but steadily drops as time goes on. Now look at the red curve representing thyroid hormone. It stays incredibly low during the early stages, which perfectly matches our first calculation where receptor occupancy was only 20%. But as we cross the dashed line into the metamorphic climax, the red curve spikes dramatically upward. This visualizes the massive flood of hormone that drives receptor occupancy to 90%, triggering the sudden and irreversible transformation of the animal. We have covered a lot of ground today, so let us summarize the key takeaways. First, Metamorphosis is a beautifully orchestrated symphony of biological destruction and creation, entirely conducted by hormones. Second, we saw that amphibians rely on thyroid hormones to drive this change, while insects depend on a delicate balance between ecdysone and juvenile hormone. Third, these chemical messengers work by binding to specific cellular receptors, acting as master switches that rewrite the organism's genetic instructions. Finally, by using mathematical models like receptor occupancy and the metamorphic index, we can precisely predict when these dramatic developmental changes will occur. You now have a complete understanding of how a caterpillar becomes a butterfly, from both a biological and mathematical perspective. Great work.